a biologist at UW River Falls, and I'll be the moderator of this session. All right, I'll introduce a speaker, and each speaker will have 6-minute talk and then 2 to 3 minutes for question and answer. And then so without further ado, I'll introduce the first speaker, which is Dr. Seth King from UW La Crosse. And hang on, sorry, I forgot to get the topic out. So Dr. King is a professor of physics at UW La Crosse, and he would talk about more than, oh, the title is on the screen, more than the sum of the parts engineering nanocomposite materials. Dr. King.
Well, thank you. They put the research thing now because we got way too excited through the quick pitch and trivia. So now we gotta bring our heart rates back down. I'm just gonna talk kind of about a project that we've been working on actually for quite a while. This was WiSys funded originally, I believe, back in 2018. And it's working with an idea of nanocomposite materials, which is a way that you can tailor dissimilar materials together in order to make it something that you want. So all of us have experience with composite materials. So material that's composed of two or more individual materials, and really more correct on this is two individual materials that have very different chemical and physical properties. So the most near and dear to your heart, even though you probably don't know it, is the idea of concrete or steel reinforced concrete. Things that have gotten more exciting, right? Carbon fiber consists of carbon fibers, but carbon fibers in and of themselves, they've got strong tensile strength, but they flex. So what you do is you put them in some sort of epoxy resin, which is sticky, but not particularly strong in the way that you want it to be. So all of these things come together. Getting closer to what I'm going to talk about, if there's athletes out there, hikers, bikers, antimicrobial clothing these days uses silver nanoparticles in order to, well, quite honestly, make it not mold when you forget it in your, gym bag. So all we're doing when we're trying to put together composite materials, right? Lighter, stronger, better, whatever the heck better means. And what better means in an engineering context? Well, it can be, it costs less. It can be, it's faster. It can be, it's blue rather than yellow. Color is a preference. So my background is in semiconductor processing, which was mentioned yesterday as something that we've kind of strayed from as a country. I would agree with that. I'm a little biased. So I used my semiconductor processing ideas in order to say, all right, what can we do? How can I make some materials that may not be useful? So my background is in transparent conductors, which all of you have literally touched today because it's on the front of your cell phone, tablet, whatever. And the term transparent conductor should hurt your brain, but that's not what this talk is about. We also work in biosensors. So what can we do for biologically compatible materials that won't kill the user? Antimicrobial surfaces, and really the sky's the limit. But anytime that I'm working on a material system for, well, general application, I try to keep it non-toxic, biocompatible, inexpensive, abundant supplies. So what we've been working on are graphene-based nanocomposites. So graphene was all the rage mid 2000s, early 2010s, and it is a single sheet of carbon. It's highly conductive. It's optically transparent, but it's got a problem of it's like a sheet of paper. It's not freestanding, and it's difficult to cover large areas. It would be like covering a window with a bunch of Post-it notes. It works, but it's not quite the result you want. So we kind of said, what can I do with graphene? And we said, let's stick stuff onto it. So a material that I've worked with a lot is zinc oxide. If you've had a multivitamin or eaten breakfast cereal, you've eaten zinc oxide this morning. Same stuff that's in sunscreen, extremely abundant. It's a wide bandgap semiconductor. It's resistance to all sorts of different things, non-toxic. And we always think of it as this white powder, but if you zoom in enough, it looks like a single crystal. So what we really did is we'd say, let's take our graphene and let's just go ahead and put a chunk of zinc oxide on it. So now what I've done is I've taken a material that absorbs light, put it onto something that's transparent. So if you think of what happens in a solar cell, I can get an electron out of here, I can dump it into the graphene, and I can make it conduct. Okay, great, awesome idea. How do we make it? Well, we figure things out. So we wanted a real simple synthetic method. So what we did is we start with what's called graphene platelets, which are basically just chunks of highly pure graphite. We sonicate them in usually water, and we put in some sort of precursor for our functional zinc. If I do zinc oxide, we use a zinc acetate, but we can do all sorts of different things. Then we say, all right, let's sonicate it, let's dehydrate it, let's homogenize it, grind it up, and then let's throw it into a really hot furnace so that the zinc acetate decomposes, or sorry, oxalate, I don't know why I'm saying acetate, decomposes, and I end up with a black powder. Well, this black powder has zinc oxide glommed onto that piece of graphene. And what we found we can do is using thermodynamics. So this is a petri dish of water. And by using magic, thermodynamics is magic. If you can see this kind of darker region, that is a film that we've been able to thermodynamically stitch together. This particular one is about 5 centimeters by 3 centimeters, but we've made these up to, well, using a 12 by 12 cake pan, highly technical, expensive lab equipment. So we've got a thin film that we can then support and put onto whatever you want. So, you know, what have we learned? We've got a lot of powder, easy to synthesize. We've studied all sorts of different properties. These self-assembled films are really what we're looking at right now, because you can take a forceps, pick them out of the water, put them onto any substrate you want, or you can what's called float cast it, highly technical term for take a piece of silicon or glass, put it under the water, pick it up. And then what we're doing is we're changing out zinc oxide for all sorts of different oxides. And then the current thing that my collaborator is working on is pick a chalcogenide, dichalcogenide if you want to be technical, and see what happens with that. This is an interesting process because like these molybdenum disulfide and tungsten disulfide are two-dimensional materials. So we just kind of stack them on top of each other. That's an ongoing process. So there's my collaborators and former students. Everyone here has graduated and gone off to bigger and better things. But always have to credit the people that actually do the work. I just write grants and stand up here and talk at you people. So thank you very much. If there's any questions, I would be happy to answer them to, well, the best I can.
Any questions? There's a question over there.
My question had to do with your first graphic with the, you showed basically the graphene.
Right.
Okay.
Like this one?
Yeah, graphene, those carbon atoms have lots more bonds available than they're showing.
Yes.
So how do you keep the graphene pure before you can work with it?
So that's actually a really, really good question. And it took a little experimentation. The simple answer is brute force. So when we do that sonication step, our graphene that we get, it's called graphene platelets. So just think of a bunch of sheets of graphene stuck together. So you're right, there's pi bonds that come out of the plane in this case that want to bond together. So we just hit it with very, very high frequency sound waves, rip it apart. So those pi bonds, they can have the problem of coming back together and restacking. And we have seen some where we have like 2 to 5 graphene layers, but it's also because of those pi bonds that allow us to do this, because it's this conjugated pi bond that comes out. So the zinc oxide is very, very happy to nucleate because carbon, oxygen really like each other. So that's a technical science term for those not in the know. And then once you've nucleated an oxygen layer, the zinc oxide just is very, very happy to assemble. So we do have some problems with thicker than one layer, not a lot, but it is something that we have really just overcome with brute force.
Okay, now due to the time constraints, sorry, we have to move on to the second speaker. If anyone has a question, I encourage you to ask Dr. King. All right, thank you. All right, we're gonna move on to the second talk, which will be very different topic. This talk given by Casey Pelpicki. No. He's from UW River Falls Department of Music, and he's gonna talk about video game music in academia. I'm sure this is a very interesting topic to young kind of college students. Welcome.
Well, on a very different topic, hello, I'm Dr. Casey Pelvicki. I'm a professor of music at the University of Wisconsin in River Falls. I teach music composition and music theory, and I have an expertise in screen scoring, which is something that we're seeing more and more in universities. It's basically scoring music for film, television, video games, and other visual media. It also involves sound design, which is sound effects, dialogue, things like that. But today I'd like to talk to you with the limited time we have about video game music as a topic of study in higher education and in academia. In general, I'm currently in the process right now of finishing my second textbook. This textbook is on an introduction to video game music through Kendall Hunt Publishing. The publishers are jumping at this topic right now because it's a very attractive field of study. And I mean, the young people love it. It's video games, right? I mean, just putting this up here on the board, I got all your attention right away. Like, what? I know what those are, right? I mean, you're just drawn to it right away, and all my students are too. And it's a really great topic. So I'm hoping that this will lead to everyone rethinking the value and the academic merit of video game music as a topic of study, a serious topic of study in academia. Okay, so it's up here a little bit. I'm a little bit taller. The problem that we're facing with this endeavor, the first one is video game music it has no place in higher education. That's a common sentiment. You'd actually be surprised. The common misconception here is also video games and their music are cheap forms of entertainment and are not worthy of serious study in academic institutions. So these are just some general things that, general speakings that I've heard along the way is video game music is not meriting. Only classical music should be studied in higher education. And of course, you'll never get a real job in that industry, okay? So I'm out to prove this wrong, okay? So my approach, let me pull up my notes here, sorry. There's a lot of notes. Okay, so starting off really quick. There's been congressional hearings, the American congressional hearings on violence in video games led by Senator Joe Lieberman in the early 90s, led many to believe that video games are a negative activity and that it corrupts young minds and distracts them from more important things in life. So of course, these hearings were unsuccessful in proving that violence in video games causes violent behavior in adolescence. That was sort of their whole goal. However, the hearings were effective in getting out the message, the negative message of video games as an activity even. So many have basically dismissed their merit value and led to those previous issues that I was discussing. So there's other prominent music critics that have lived. Theodore Adorno, if anyone knows who that is, he's notoriously he bashed film music for many, many years, saying it was, it dumbs down the masses. Quote, it dumbs the masses. While he wasn't alive during the inception of video games and video game music, I imagine the sentiment would be the same. I also conducted a survey. I conducted a survey of 40 music scholars, mostly tenured professors from various disciplines, and most of them, the majority of them believe that music in academia should be focused on traditional and modernist classical concert music. Okay, so if you know what modernist classical music is, it's if you guys know who John Cage is, there's a piece called 4 minutes and 33 seconds that's really famous, where he sits at a piano for 4 minutes and 33 seconds in complete silence. and that's the piece he gets up and bows. So that kind of music, but also traditional music, Bach to Brahms is what they feel is the more academically worthy. 35 of them stated that there should be room for both, though. Okay, so that's promising. A lot of it's because 33 out of 40 of them stated that they don't regularly play video games or really have much knowledge of video game music in general. So there's another problem too. It's awareness, bringing awareness to this topic. 1 second here. So to address the problem, I performed a series of score analyses. And if you don't know what that is, it's where I go through a large music score and analyze every note. It's a lot of fun. Examined several books and journals on the compositional process of video game music, which they're becoming more in abundance. Oops. And I attend regularly this Game Sound Con in Los Angeles every year. I bring some students, and you'll find that there's actually a lot of people that are into this. This industry is actually pretty massive. My discoveries in general are video game music shares the same characteristics as classic, traditional, modernist concert music. They both have melody, harmony, rhythm, timbre, orchestration. Video game music is actually more complex, far more complex than traditional classical music, because there is a need to understand the technology alone. That's an entire degree, really, understanding the technology that goes into this, but also understanding acoustic and electronic music, music theory, and repertoire. And also an additional aspect is an understanding of narrative and interactive music, okay? So what's interactive music? You can kind of get an idea. This is what it kind of looks like on the computer. But video game music is immersive and interactive. Interactive music is essentially music that attempts to adapt to the choices and progress that a player makes in the game, okay? If I choose to go left, the music will play something. If I choose to go right, it'll be different music, okay? It's music that adapts to tailored to your experience. Okay, there's a plethora of jobs and career paths in video game music and audio. The Game Sound Con has proven to me that there are jobs abundant. And of course, my application of this is UWRF has allowed me to teach courses in this topic, which has been excellent. Getting that opportunity, the upper administration has been very supportive. I was awarded the Manley Olson Grant Award to research the topic. Esports has been helped to usher in more and more of this topic. And of course, I'm working on a certificate program that's in the works for summer of 2025 in this field. Finally, really quick, the impact in academia is it's an attractive discipline. It increases enrollments, which all my classes are totally booked every semester. It's a cutting edge field of study, and there's interdisciplinary opportunities everywhere. Society facilitates change in public perceptions of video games and video game music. There's public performances now in popular venues. And then, of course, the experience of playing a game in general gets better if we can study it more. And then students, my students get opportunities to build a demo reel for job applications, a variety of disciplines, and they are collaborating with different departments. So yeah, there you go. And I'd be happy to take any questions.
There's a question in the middle.
An interesting, presentation, Professor Pelbeki, and I can relate. My sons play video games, Pikmin 4, Mario Brothers, and the music that I hear. I sometimes sit with them, play with them. It's very complex. You're spot on there. Anyway, I'm curious about the disciplines, collaborations. Can you?
Absolutely. So the disciplines, obviously, computer science and video game design is one of the disciplines that we try to interact with and collaborate with. There's also everything from acoustics, which is sort of a physics topic. Art in general, sound design, which is voice acting, and Foley artists, which is like creating sound effects in like a room, and then also technology. Computer science, just understanding the technology of recording music and creating it, and then like how to distribute it. So there's a lot of different areas, the business side of it too. I could go on and on about all the disciplines that are involved in this, but yeah, it's very global in a lot of ways.
We just have one more question. Right.
Dr. Pelbeki, I have a question which is a little off than video games, but I have a question related to rap. So rap is considered as one of the genres which has a little bit violence involved in it, but at the same time, the positive effect of it is increase in self-worthiness. So what's your take on that? Because it just explores both the sides of the coin.
Yeah, I may be biased because I'm a huge rap fan, actually, believe it or not. So I think it's fairly disparaging that people judge it by its cover a lot of times. You're actually seeing it more in video games, though. We're actually seeing some things like Parappa the Rappa was a game back in the late 90s that was really popular that introduced rap into video games, and we're starting to see it more and more. But I could tell you that, yeah, I think it's just a miseducation or a misunderstanding, misconceptions with rap and not understanding the true culture, the true art form. Because a lot of the stuff that's put in our face on MTV and on the popular radio is pretty bland and thoughtless in a lot of ways.
All right, one more question.
I'm interested in your analysis of complexity. It seemed to focus on how the music is generated, but have you looked at it from an information theoretic approach and the predictability of the music as it progresses? I know this has been a topic used to analyze classical music, I think Bach in particular, and there's connections between complexity from an information theoretic approach and the emotions that the music elicits.
Yeah.
Yeah.
I mean, this is deep in the weeds here, but I would say, yeah. I mean, complexity technology wise is how do you get the music to do what you're doing? How do you get it to change? If you're in a boss fight and you're losing, the music will change sometimes, start to become more sad or dark, right? But if you're winning, it becomes more triumphant, right? So they'll bring in different instruments and things like that to signal that. But as for like the emotional, like how we do this, there's a lot there. I mean, you talk about how pacing is one example. So like the tempo of the music versus the tempo of the action on the screen will affect how we perceive what's happening. If you're in a chase, you expect to have drums or some fast music. But if you're in a long, slow dialogue scene, you expect slower music that's more sustaining. And so it's basically in a supportive role to enhance what the narrative is giving us emotionally, right? And that's kind of the big point about this is that the narrative gives us something and we are using the music to reinforce it and enhance it further. Sometimes it's subtext where there is nothing on the narrative and the music is telling us the story too. So.
All right, thank you, Dr. Palbicki. I'm sure many of you probably have more questions, so I encourage you to talk to Dr. Belpick after the session. All right, our next speaker is Dr. David Hicks. He is a professor of bioscience at UW-Platteville, and he's going to talk about TLN1, a new algal strain with superior growth and lipid accumulation in low nitrogen. Welcome.
Great, thank you, and good morning, everybody. Thanks to WiSys for the invitation, as well as for an Ignite grant that's helped fund this. That funds the project, but also supports students involved in the research. So I'm going to talk this morning briefly about a strain of algae, microalgae, that we've been working with for a while in the lab. We work with a Chlamydomonas reinhardtii species, and the algae we're talking about is called TLN1. It's one we published on a number of years ago. The TLN stands for tolerance to low nitrogen. I'm going to talk about how when grown into low nitrogen, algae respond by increasing these triacylglycerols or tags, which one of their uses can be to be converted through a transesterification reaction into biodiesels and a potential source of a biofuels renewable energy. And so the strain we're talking about and working on grows better into low nitrogen as well as produces higher amounts of these triacylglycerides or tags. So as we all know, photosynthesis is the biochemical process that pulls CO2, carbon dioxide, out of the atmosphere through the biochemical reaction. It uses the energy inherent in sunlight to then store these molecules, carbon, in high energy reduced carbon forms, oftentimes in carbohydrates, sugars, starches for the plants, as well as in different forms of lipids, including the triacylglycerides, which again can be used from lots of biomasses or feedstock, can be converted into biodiesels, and many of them can be a drop-in energy source, renewable biofuels used in current existing engines, be it vehicles, be it construction equipment, be it farm equipment, as well as by mixing with kerosene to a certain extent, can be used in different aviation fuels. And this morning there was a reference to the sustainable aviation fuels or SAFEs. That's an area that there's a lot of interest in. So this happens in plants, it happens in trees, it happens in crop plants, corn, soybeans, but also happens in algae. Larger or smaller algae, microalgae is sometimes called, single cell algae. And again, the particular species that we tend to work with in the lab is Chlamydomonas reinhardtii, or clammy. And so I'm going to be talking more about this particular algae. Now, again, these triacylglycerides can be converted into biofuels, biodiesel, There's many sources or feedstocks that can be used for that. But one of the areas that's been looked at and considered for quite a few years is the issue of, can you grow algae to produce large amounts of it? The advantage is the single cells are basically photosynthetic factories that can produce really high levels of these biodiesels or biofuels, up to 50 to 60% of the biomass of these cells. The challenge is growing them up, generating enough. The scalability is an issue that has yet to be dealt with in a significant way. Again, there's been a lot of interest over many years, but more recently, there's been a growing interest in producing these sustainable aviation fuels or SAFEs. And one of the many stocks that are being considered for SAFEs are some of the algae as a source of biodiesel potentially. And there's a target to increase the use of these sustainable aviation fuels over the next several years, by 2030 and 2050. The goal is to significantly increase the use of these, in part because the aviation industry, 11% of the emissions from the transportation industry comes from airplanes. So this algae we work with, this or the TLN strain, we can measure these lipids using a Nile red, a very standard way to visualize as well as to quantify. Inside the algae, when you grow them in low nitrogen, they accumulate these large lipid bodies that you can see with this, excuse me, the yellow stain that can be visualized, as well as we can quantify them spectrophotometrically. So in algae, a response to a natural phenomena is when they're under stress of low nitrogen, they will start to accumulate high levels of these lipids. So it's a survival mechanism. They're kind of preparing for bad season, overwintering, by accumulating high levels of these storable high energy molecules. And so that's an advantage. And they will increase up to 100 times higher levels of these tags when grown in low nitrogen. The problem is that also dramatically reduces their growth by up to 50%. So one of the goals is to try to find variations, strains that do better under low nitrogen, yet still have these high level of these lipids that are being produced. In this strain, this TLN1 strain has a lot of these features. So when you grow under low nitrogen, this TLN1 strain will accumulate about 40 to 50% more cells in higher biomass compared to the wild type variants. And when also grown under low nitrogen, they will accumulate between 7 and 10 times higher amount of these tags. Again, can be extracted and then converted into potential biodiesel. So these are of interest. And so we're interested in this particular strain to pursue it, to figure out, will this be potentially useful as a source? So we wanted to dig deeper, and we're using a genomics and a molecular genetics strategy to investigate this. We sequenced the genomes from the strain and other progeny from the strain that had this phenotype, compared them to the wild type. When we look at that DNA sequence, we have identified 2 different candidate genes that have genetic sequence changes that might be the cause for this TLN phenotype. So from that, the 2 we're looking at, there's the TAB2 gene, which is a known characterized gene. It's a regulator of protein synthesis. Another one is a predicted zinc finger transcription factor, also a regulator that may control other genes. So fortuitously, the TAB2, there are 3 known mutants that have been available for quite a few years that have mutations in that TAB2 gene. So we've obtained those. We're investigating the phenotype of those. And we sequenced them first to confirm they actually have sequence changes in those genes. And then we investigated the phenotype for that TLN trait. Do they grow better under low nitrogen? Do they produce higher amounts of these triglycerides? And the answer is they do. The F12, F14, F20 strains on this table, they grow similarly to what the TLN strain looks like. So it appears that the mutation, the TAB2 gene, is likely candidates for this. We want to go further with this, and we want to generate some new mutants using CRISPR-Cas9 gene editing. And our ultimate goal is, if these changes do cause this increase in triglycerides, can we move them into other plant species or algal species? Our first next step would be to move them into more industrially available strains such as chlorella. They grow better. They've been used industrially since the 1940s for either food products or other sources. So with that, I'm going to stop, and I want to acknowledge again some of the students that have been involved in this project over the number of years, including Nicholas Winter, who gave one of the quick pitches this morning, gave a poster yesterday, as well as Andrew Lawson. And again, let me acknowledge WiSys for funding through the Ignite. Thank you.
Question? So you mentioned that there are other plants that have been used in biofuel. And then you mentioned the advantage of using this microalgae. Are there any algae that can be used as well?
Oh, there's quite a few.
Oh.
There's been historically a lot of interest in looking at a variety of different species and strains of algae. Chlorella is usually high on the list for that. There's also some photosynthetic bacteria that have been looked at. The reason we've started with Chlamydomonas is it's probably one of the better studied of the green algae as far as its genetics, the biology, the biochemistry. So it's a great starting organism to investigate the biology. It's probably not the best final organism, but there is a long list. It also depends upon where would these be grown. In certain environments, certain species would do better based upon the environment, temperature, climatic factors as well. So yes, there's a huge variety of those. In different regions, probably different algae might be better choices. Good question.
All right. Any other question? Oh, there's a question in the back corner.
Thanks so much for your presentation. I'm curious if you've looked at the Bioenergy Technologies Office at DOE for funding, because they have been interested in algae, especially they're looking more at the creation of intermediates that then could be maybe taken to petroleum refineries to be co-processed with fossil fuels for renewable diesel as opposed to biodiesel from trans-esterification. So I'm just curious how much you've explored that as opportunities.
Yeah, no, it's an excellent question. And I, your comments this morning, and obviously listening to what you were talking about, you mentioned the safes earlier. And as you mentioned, DOE has been really interested in this for decades. Again, this is not a new topic. It's not a new idea. I don't have as much in-depth knowledge, and I've looked a little bit, but not enough to be able to comment in an expert form in that. But absolutely, there's a lot of different sources, different feedstocks, and different molecules from algae and plants that are being looked at. And this is obviously just one of the possibilities. But yeah, it's a great question. I don't have a really exact response, but I do appreciate that. Your comments earlier about this whole area. Thank you.
Okay, let's thank Dr. Hicks again. Thank you. All right, our next talk will be given by Dr. Oscar. I'm sorry if I pronounce it right. Yavuz Setin. He's associate professor of physics at UW Whitewater, and he would talk about workforce development through undergraduate research in an interdisciplinary travel study course. That's interesting. All right, welcome.
Hello, everyone. This is a little bit like a follow-up from our panel yesterday, where we were discussing high practices. And I mentioned about this class that I've been running with my colleague, Jup Bhattacharya, from the Geology, Geography, and Environmental Science. So I'll try to give you a little bit more idea about what this travel study is and what kind of projects the students do and what kind of skills they acquire from this course. So first of all, travel studies are known to be a high impact practice classes. The students get to have the international travel, and also there's the cultural immersion part. Now, especially when it comes to more like the benefits to the students, they are definitely accelerated high impact practices. There's a transformative learning part. It's a lifelong friendship they can form if they work on common projects, and it's fun. Now, one of the difficulties of travel study class is that balancing the academic work with the travel part. You don't want to turn into a touristic trip or a selfie session. The excitement of travel and new experiences can distract the students. Usually when students travel to a different country, they can feel like a kid in a candy store. There's so many distractions. So as faculty, we still try to keep them focused on their projects. At the same time, they have fun learning new things. And other objective that we have is how they can acquire skills for the workforce development. So this is from an image in the Lava Center in Iceland. By the way, our travel study class is only to Iceland. And why we picked Iceland? Because it's safe. I mean, don't worry about the volcanoes. They don't happen everywhere. Geologically, it's safe. It's one of the safest places in the world, actually. So this is from a lava center, and this is from our second cohort of students traveled there. So I'll show you another one. And here you can see there's a student here. So this student is right now teaching at Lava Center in Iceland right now. So that was a pretty fast transformation. So students came with us last year and basically visited Lava, not last year, 2 years ago. And later he was so impressed with Iceland that he made communications and he basically got an internship at the Lava Center in Iceland. And right now he's educating the group of visiting students in that place. Of course, this travel study class is open to all the students. It's not only like physics or geology, it's open. It's a general lab class. And the trick is like, how do we incorporate some of the generalized skills that can help them in their future lives? So a little bit details that can be boring. It's a lab-based class. It's pretty unique because it's not very common to see travel study classes with a lab-based component. Why lab-based? Because Iceland is an open field lab, especially for geologists. Everywhere is in the accelerated frame of transformation. It's 4 units. We ran it in 2018, 22, and 24. There's a little gap in between because of COVID. We meet twice a week with the students. And from the first day on, we ask them, what is your project going to be? Have you thought about your project? So we try to remind them about their projects. We also offer a little bit of labs because there are a lot of non-science students in our class. And every student has to do a project, no matter what. That's the requirement of our class. So we ask the students to bring either their projects or we ask them to form groups, and we can offer them from a variety of projects that we have. And it's also inclusive. We have all majors, freshmen through graduating seniors. And then we have like students coming from different campuses like UW Rock. We also have students who are non-traditional. So we are open to all kinds of students and also students with disabilities. So this is our cohort in 2018, just to give you a little bit like nice scenic view in the background. That's my collaborator, Duke. And that's 2022. That was a sustainable farm visit in the background in Iceland. And that's from this year, just in May. So some of the discussions, like some of the things, first of all, the students, what do they learn? These are the students, if you think about like, we have never done projects in their lives. They don't even know what poster means. So we're kind of like immersing them in the life of poster presentations, projects, proposals. So basically students have to go through all these steps, even though they have never done it before. And at the end, we also ask them for their commitment for dissemination. That's typically poster presentations at the undergraduate research days that happen at our school, or if they make up to the next level, they can present at other venues like Geological Society of America or other places. And they also present their posters before the trip, after the trip, when they come back, and also in Iceland too. So for example, this one is a presentation in Iceland, very remote place surrounded by hills and mountains and glaciers. Very hard to find the technology, but made it work. Students measure everywhere. This is the EMF study of a student doing in a graveyard. And this is in a bus stop in Iceland. They're very dedicated to work, as you can see in different environments. And here is a collaborative work that we do with our local guys over there. So some of the common things the students learn, they improve their team player skills. They're working on interdisciplinary projects, and they also acquire some of the soft and technical skills. Some other projects, students like to have fun. They are not fishing, they're actually sampling water here in the glacier. I believe it's Vatnajökull. And some of the equipment we do, and I'll just show you an example. So these are some of the projects that our students are doing and some of the equipment that we take. That's one of the showcase projects. a student from geology geography designed this geyser setup. It can measure the pressure and the temperature at the same time. So he all learned by himself how to program a Raspberry Pi using Python, added all the sensors, and now we will be using this, actually, not me, my colleague will be using this in her classes in the intro to geology, just to give an idea about what geyser is and what kind of things you can measure. So I realize my time is up, but meanwhile, I'll show you a couple more pictures and some of the student reflections and questions if you have. After a long day here. That's a nice one. With another non-traditional student, we're analyzing from the samples in SCM. So I like this comment about that. The holistic learning approach. And of course, students also do some avatar projects on us. That's their
Okay.
time.
Any questions? Question in the middle.
Yeah, thank you, Oz. And I think we met virtually through Second Nature.
Yes,
I
I
now
remember
looking, seeing Project's name there and your name together. Anyway, it's great, this travel study initiative. My curiosity is, I think, I don't know whether I missed that part. How you ensure this travel of interdisciplinary, like, is it like a, and also in the semester, looks like you'd have a poster presentation before, at the beginning of the semester, then middle, you take the students to Iceland, and they come back and come up with the second poster presentation, right? So basically, I'm just curious, how, is it like some sort of grant, or the students come up with their own budget, or, just curious.
Yeah, so everything starts with the beginning of the semester. So we have to first recruit students, find them. And basically the first day, we start with, okay, what's your proposal? It starts with that. And then they write a proposal to ask for funding for their equipment. And our undergraduate research program funds at a limited budget for some of their equipment. So they can, if their proposal is good enough, they can get funding, or we provide some of the equipment to them. And then they do the first part of presentation, typically in the first, second, or third week of the class. Then if there's time, they can do a second presentation, then they do it in Iceland. And then after they come back, they also do at the undergraduate research day.
Is this program only open for UW Whitewater or the UW campuses?
Anybody can apply that program, but we don't advertise it very much. It's mostly for the Whitewater students primarily, and also for UW-Rock, but technically anybody actually can take it from other campuses as well.
And how long do they stay in Iceland?
Typically like 7 days,
Oh,
7 to 8
okay,
days.
gotcha. So most work are done here.
Right.
Okay,
Most of the
gotcha.
field work is done in Iceland. Sampling.
Okay. Any other question? All right, if not, that's thank you. Doctor again. Oh. All right, now we are moving on to our last talk, and this will be given by Dr. Mohammed Rabbani. Wow, it's like a cheering. All right, Dr. Rabbani is an associate professor of chemistry at UW-Platteville, and he's going to tell us about the modification of cotton fabrics for antimicrobial applications. Welcome.
Thank you, Dr. Huang, for your introduction. First of all, I would like to thank WiSys for organizing this wonderful event. And this is really great to know and talk to professors and students coming from different universities. It's really great. I'm going to talk today about an ongoing project that is modification of cotton fabrics and potentially for future application in antibacterial study. We just recently had a terrible COVID-19 pandemic, and more than 15 million people died worldwide only in 2020 and 2021. And this is because of viral attack followed by bacterial infection. So in order to reduce the spread of infectious disease, at one point, face mask was mandatory, and it is highly recommended to use disinfectant. So alcohols like ethanol or isopropanols, those are commonly used ingredients in many disinfectants. So here is a alcohol spray commercially available, and it is 75% alcohol, and it can kill more than 99% germs. However, there are some challenges to use alcohol to kill these germs. Because alcohols evaporate very quickly, you need enough concentration to make sure bacteria or viruses are killed before evaporate alcohols, and germs can develop again on the clean surface. So the possible solution is how to hold the alcohol molecules on the surface rather than evaporating. So that is my research, how to chemically modify the cotton fabric and hold the alcohol molecules. So who will get benefit from this? We can use that modified fabrics to make face mask. It can be used for surface cleaning cloths, germ-free clean room in hospitals, gauze cloths like in hospitals, and particularly for infected carols here. So we can use, this is particularly really important. And according to US farm data, in Wisconsin only, we have around 17,000 dairy farmers. So if we can come up with a solution which can inhibit the growth of bacteria, then it can make a big difference in not only in Wisconsin, but also across this country. So how to work alcohol in killing bacteria or germs? So I'm going to talk very quickly. Alcohol has hydroxyl group that we call the water-loving part, and it also has carbon chain, which we call the fat-loving part. So both fat-loving part and water-loving part going to attack the cell membrane, and then it denature and coagulate the proteins and finally kills the germs. So that is the way how alcohol works. So there is a recent report which shows that ethanol and isopropanol, both are alcohol, inactivate the coronavirus on the solid surface. So knowing that, now how we can bind alcohols to the cotton. So this is the chemical structure of cotton, and this is a polymer cellulose in which 5-member sugar molecules are connected through ether linkage, we call. But the characteristic feature of this is it has hydroxyl group, pendant hydroxyl group. This is the place where we can modify, we can do modification there. So we did an experiment reacting here with sodium chloroacetate. This is another compound, and to connect that carboxylic acid. So once we get it, so then we can use this particular part to do the next connection. But in order to confirm whether it is connected or not, we took infrared spectrum. So the red spectrum is for the cotton fabric without chemical modification, and the blue spectrum is after modification. If you're a little bit aware of if you know IR spectrum, this is the particular peaks around here. There is the characteristics of carbon-hydrogen stretching. It's everywhere in the molecule, and O is stretching here. The cotton fabric itself has OH, so that is shown here. But when the modification is done, you can see it is stronger and extended and wider. And that is another characteristics of hydroxyl group, which is connected to carboxylic acid. So the last characteristics of carbonyl group in carboxylic acid is observed here. So from this study, we can say the connection was done perfectly. So the next step, what we are working now is to connect a diol, one of the hydroxyl group, to here through a well-known reaction we call esterification. The end product we are expecting like this, and that part is exactly similar to an isopropanol, which is used in a disinfectant. So we're working on that. So the first step, what we are doing, because if you think about this is a cotton, we cannot take NMR, we cannot confirm the connection was done or not. So what we do, we selected a small molecule like ethanol and trimesic acid we call. These are isolated molecule. We just connect each other through esterification, same reaction, and we can isolate the product, and we took NMR. So this NMR spectrum confirmed the formation of the ester bond. This hydrogen is observed here. This peak is for this CS2, and CS3 is observed here. So this confirmed the reaction can be possible. Now students are working on the actual molecule, how to connect this one at first to this and confirm it. And then finally, we're gonna connect that molecule to the cotton fabrics. So that is our overall plan, and we are working on that. So once it is done, then our collaborator, Dr. Levinston, he is doing bacterial study. So I will send the sample to him. But at first, I have to make sure this is done. And Faith, he is actually doing modification as well as bacterial study. And Ryan and Erica, they're working on modification of cotton fabrics. Of course, we're thankful to EW Platteville for supporting undergraduate research. And WiSys, of course, without funding from WiSys, it is difficult to do research for undergraduate. So thank you very much.
What do you want? Or you can use mine. Is it on?
It's on now.
Try it. No, it's not.
How does the modification change the properties of the fabric? Would you still be able to use it as a bandage and a mask? And then if you used it as a mask, would you then be inhaling isopropanol? Would that cause other dangers or issues?
Properties means, you mean physical properties or?
Yeah, the physical properties, just being able to make it into a mask or use it as a bandage instead of if it makes it hard all of a sudden and it won't bend, or if it makes it really abrasive.
Okay, it's a good question. The first step, what we did, the modification to connect the carboxylic acid. I don't have the image here. I have another slide somewhere. So there is no difference. Basically, we took the picture, and apparently both looks like physically exactly same, similar appearance. So as I mentioned, we are still working on to get the final stage. And when we will connect the final stage, I don't know what will happen, but we are expecting the first step. We are doing the reaction under very strong basic condition using sodium hydroxide for a longer time period. And it didn't change the physical appearance. So we're expecting in the next step, it will also stay as it is. Thank you. It's a good question.
I got one.
Yes.
So when you talked about how you were adding that molecule to the appendage OH of that cotton, is it possible that it could add to a different OH that was seen in there? And then if so, would it have any different impacts to it? Because I know that changing that carbon structure could change its function.
So you're talking the first step or
Yeah.
second step?
The first step.
So, okay. So there is a difference here. You can see this OH is CS2OH. But if you are talking about this one, this is directly connected to the ring. So we anticipate that this is connected to the longer chain. It is a little bit sterically a little easier. And this is known that it's a alkane chain. So reaction is supposed to be happen here. Thank you.
All right, we have one more question.
Yes.
How many, like, do you think this will have to happen at every OH along that chain, or is it like only 50% or something like that?
Yeah, that's another good question there. How much we can connect, right? So that is another, first of all, if we know that we can do it, then we can extend the time period. Like now, I think we're using 30 minutes reaction time. So we're gonna go like an hour reaction time. Probably I'll go like overnight reaction time, extended time period to give enough time to see if there is any difference or not. So we still need to do that reaction. It's a very good question. So we really want to make sure that we maximize the concentration of alcoholic group connected to the fabric. Yes.
All right, thank you very much. And let's
you.
thank all the speakers for the wonderful section. And thank you all for being here, all right, to support this symposium. Now I'm gonna pass the micro to. Oh, you want to tell them?
Now we have lunch, so the lunch is outside. Please help yourself, and then we'll come back in here for some closing remarks. Thank you.
Thank you.
Thank you so much.
You're welcome. Thank you.